Hi, my name is Jessie Turner, and I'm a graduate student at the Virginia Institute of Marine Science, or VIMS. I study water clarity, how clear the water is, and how light interacts with the substances in the water. Today, I'll be talking about Beer's law, which describes how light interacts with liquids and affects their clarity. This talk was originally going to be presented during our Scientist Walks into a Bar event in March, which was canceled for social distancing. So I'm going to stick with the theme of drinking beer together yet apart and use beer as an analogy to help us understand water clarity. So if you want to, now's a good time to pause the video and get a beverage of your choice. Today I'll be talking about water clarity in the Chesapeake Bay using beer as an analogy. Weirdly enough, Beer's law all started with wine. Beer's law was discovered by a Frenchman named Pierre Bouguer while he was on vacation in Portugal, gazing into the depths of his wine glass and seeing that light disappeared as it went into the wine. Later on, this idea was rediscovered by a guy named Lambert, and then by the namesake August Beer, who named the law after himself. So now it's known as Beer's law. So what is Beer's law? It tells us that how much light gets through a liquid depends on the concentration of stuff in the liquid and the distance you're measuring over. What's the stuff? Well, in Chesapeake Bay, the stuff in the water might be little particles, tiny pieces of dirt, microscopic plants, or dissolved substances. In beer, the stuff might be yeast, hops, or dissolved things like sugar or tannins from the brewing process. When we measure clarity by way of Beer's law in Chesapeake Bay, we measure it in something called a cuvette, represented by the green liquid in the little glass on the screen. We use a spectrophotometer, a machine that shoots a light beam through the cuvette and measures how much light gets to the other side. The more light gets through, the clearer the liquid is, and the clearer it looks to your eye, too. The distance is the distance through the container. So this could be the distance across your beer glass or down through your beer glass. Another way we measure clarity in Chesapeake Bay is something called a Secchi disc, just a black and white disc. And as you lower it through the water, the depth at which you can't see it anymore, the depth at which it disappears, is the Secchi depth. So you can try this at home. You can print out a black and white Secchi disc and put some tape over it and lower it through a liquid to see how deep you can still see it, and that's the Secchi depth. So how does the stuff in a liquid actually keep the light from getting through? When light hits water or beer, it can either get scattered or it can get absorbed. Things that absorb light are dissolved substances called dissolved organic matter in Chesapeake Bay. Basically, the types of things we find in liquids like wine, tea, coffee, tannins, dissolved colored substances that seep out of whatever it is you're brewing. And things that scatter light are particles. Particles are plankton cells, mineral sediments, that's dirt, or detritus, that's dead stuff, in Chesapeake Bay, or the types of things we would find in liquids like milk, cocoa, or really pulpy orange juice. In beer, particles like hops or yeast. In terms of beer, a dark beer like this one on the left has high absorption and high scattering, or has a lot of dissolved materials and a lot of particles. A hazy IPA, or something that's cloudy but light colored, might look really cloudy but still bright colored because it has low absorption but high scattering, or lots of particles. And then last, you have your clearest beer, or ciders, that have low absorption and low scattering, or not a lot of dissolved substances and not a lot of particles. In the bay, this looks like, from left to right, you have your darkest water, where there's a lot of absorption and a lot of scattering. Then there's cloudy water that's still really bright, or doesn't have much absorption, but it does have a lot of scattering, a lot of tiny particles scattering light. And then you have your very clearest water, which doesn't have a lot of absorption, and it doesn't have a lot of scattering either. And that's very clear water. So take a moment, maybe if you're enjoying a glass of beer or a mug of tea, and think about the clarity of that liquid. How much light is getting through it? How much light is being absorbed versus scattered? And while you sip, maybe think about the last time you were in the bay or in some kind of ocean water, anywhere in the world. What did it look like? Was it dark? Was it bright? Could you see the bottom? How much light was getting through? Another thing to remember is that clarity is not the same thing as quality. Clarity is just how much light gets through. But quality depends on things like temperature, oxygen for the fish and animals, how many nutrients are there, or the chemistry, and how much plankton or algae is growing. Similarly, beer clarity just depends on how much light gets through. But beer quality depends on things like temperature, carbon dioxide bubbles, is it cold, is it fresh, what's your personal preference? So they're not the same thing. So why should you care about water clarity? Well, water clarity matters to everything in the water, so all of the plants and animals in the water. A few examples are little plants like phytoplankton need light to grow, and they drive the health of the whole underwater ecosystem. And bigger plants like seagrasses are really sensitive to water clarity. And those seagrasses in the Chesapeake Bay or other shallow environments stabilize the bottom and provide habitat for young fish and crabs. And fish depend on water clarity to see their prey. And because of that, some fishermen depend on water clarity for their catch so the fish can see the bait. In my research, I study water clarity change over time in the Chesapeake Bay, including how clear the water is, how much light gets through, and how light is interacting with the materials in the water, such as how much of the suspended particles in the bay are inorganic matter like dirt versus organic matter like plants and dead things, because that affects how the particles absorb and scatter light, like we just looked at. I'm finding that even though the bay seems to be getting cloudier in terms of Secchi depths, even so, there's either no change or an increase or a positive change in how much light is available to seagrasses. So just as much light is available over time. And that has to do with the types of particles in the water. So this would look something like the cloudy but bright scenario that I showed earlier. If the bay is getting cloudier, it's not getting any less bright, which is good news for the seagrasses. Thanks for watching. Here's my email, and I'm here virtually if you have any questions about water clarity. Also, you can subscribe to the eTidings newsletter if you want to know more about the science we do here at VIMS. Cheers!